So today you're going to show us your work from your recent paper, which was published last month. And we are looking forward.
Okay, thank you so much, Erdich, and thank you everyone for joining me here today. We're talking today about our paper that we published, like you said, last month in Nature Communications on the lipidomic and biophysical homeostasis of mammalian membranes. The Levental lab studies the overall complexity of mammalian membranes. We are broadly interested in how the chemical composition and the detailed lipidome affects biophysical properties like lipid packing and fluidity to drive the overall function of membranes and ultimately affect cellular physiology by changing signaling at and through the membranes. Recent advances in mass spectrometry have identified that cell membranes are comprised of hundreds of various lipids species, and lipids behave in a collective manner. This means that they interact together, and it is their collective overall biophysical properties and chemical composition which influence cellular physiology. In non-thermoregulating organisms, the most common challenge to membrane properties comes from temperature variations. Cold temperatures reduce the motion of hydrophobic lipid tails, which forces the lipids closer together, dramatically reducing the fluidity of the membrane. This is obviously disruptive for the biochemistry of life, which relies on precise, fine-tuned interactions and conformational changes. In response, many organisms have converged on a homeostatic strategy wherein tightly packing lipids are replaced by non-packing ones to maintain the physical and functional state of the membrane. This remarkable adaptiveness has been observed over the last 50 years across organisms from single-cell prokaryotes up to reptiles and fish. But if you're interested in mammalian membranes like we are, does this really matter? In general, mammals and other warm-blooded animals are not subject to large-scale variations in body temperature. Thus, there's been relatively little investigation of homeostatic membrane responsiveness in such organisms. However, mammalian membrane homeostasis is extensively challenged by dietary inputs. Many nutritional factors and supplements have direct effect on lipids, but a clear study of how they affect the lipidome and their specific mechanism of action is lacking. It is known that excess cholesterol and saturated fat consumption is implicated as an exacerbating and potentially causative factor in cardiovascular diseases, while dietary supplementation with fish oil and omega-3 PUFAs have many beneficial effects. But how and why these lipids affect cellular physiology is unknown. One reason for why fats specifically affect physiology and cellular behavior is that they are directly incorporated into our membrane lipids. Thus, dietary lipids have major impacts on membrane compositions in vivo, and these perturbations presumably must be buffered to maintain cellular functionality. In this paper that we're talking about here, we directly evaluated the hypothesis that mammalian membranes homeostatically adapt to dietary inputs. We can recapitulate dietary alterations in cell culture using model cell lines. Here we use rat basophilic leukemia cells, or RBLs, and we supplemented their medium with either DHA or arachidonic acid as a bovine serum albumin complex fatty acid, which is the main source of much of the cell culture membrane lipid components. We found that these cells specifically take up this fatty acids and incorporate it into their membrane lipidomes. We then wanted to look at how specific this was to a variety of different fatty acids. We found that polyunsaturated fatty acids were specifically taken up by the cells and incorporated into their membrane lipids, whereas oleic and palmitic acid were not. We speculate that this could be due to sufficient amounts of oleic acid and palmitic acid, and that the concentrations used here have no additional effects. On the other hand, omega-3 and omega-6 PUFAs cannot be made by cells, and the cells are limited by what's available in the medium, which in bovine serum is little to none. Thus, at physiological appropriate concentrations, there's a specific uptake of PUFAs. Further, and perhaps more interestingly, we noticed right away that there was not just an increase in the DHA-containing lipids in the cells, but rather that there appeared to be an overall remodeling of the entire cellular lipidome. This figure shows a heat map of all the changes to the overall cellular lipidome of either the crude membranes or the plasma membranes of cells, which were fed DHA in the cell culture medium. Lipids that go up are shown in red, lipids that go down are shown in blue, and I hope you can see that many lipids go up and many lipids go down. And specifically, the ones that are going down don't appear to be a random downregulation of the lipids in which DHA is not entering, and there are many lipids going up that are not just lipids containing DHA. This then led us to ask, how can the addition of one fatty acid to the cell's diet induce such drastic overall changes to a cell's lipidome? So looking closer at the cell's lipids unsaturation changes, we see increases in lipids containing the supplemented fatty acid. Cells which were fed arachidonate now have lipids which have unsaturations containing either 4 or 5 double bonds. Cells which are fed DHA have lipids containing either 6 or more than 6 unsaturations. But interestingly, what we also see is that there's a significant increase in lipids which are fully saturated, and there's a significant decrease in lipids which have 2 or 3 double bonds go down. And so this suggests, and since we saw this in many different cell types, that all of the cell types we've looked at thus far, RBLs, neurons, mesenchymal stem cells, and CHO cells, we always see the general trend of fully saturated lipids go up and polyunsaturated lipids go down. And this suggests that this is a general mechanism for at least cell culture cells. But to test whether this occurred in animals in which we can modulate the diet, we fed mice a diet high in fish oil or corn oil. To our surprise, we found that lipids which are isolated from their heart had a huge increase in the amount of omega-3 containing lipids. 30% of all heart membrane glycerophospholipids had an omega-3 PUFA as one of their acyl chains. And similar to the in vitro results, we again saw that there's an increase in saturated lipids and a decrease in the polyunsaturated lipids. The similarity of these responses between freshly isolated tissues and cell cultured lines show that comprehensive lipidomic remodeling in response to exogenous PUFAs is a general phenomenon for mammalian cells. We next want to determine the temporal dynamics of this time course of incorporation of PUFAs and the subsequent lipidomic remodeling. We found that DHA is rapidly incorporated into membrane lipids with a half-life of about 4 hours. And at the same time that DHA is being incorporated, saturated lipids are also increasing and diunsaturated lipids are decreasing, suggesting there's nearly concomitant lipidomic remodeling with the addition of a PUFA to the cell culture medium. So, but why do cells do this? Lipid saturation directly affects membrane biophysical properties, as we discussed earlier, such as membrane fluidity or packing. Membranes that are tightly packed are robust and strong, but they're largely immobile. This can prohibit the dynamics which are required for life. Conversely, membranes that are too loose are unable to perform their crucial role as a selective barrier. Thus, life is only possible within this narrow range of optimal membrane properties. Sustained deviations from these membrane properties are harmful, if not fatal, to the cells. So how do living membranes maintain these properties in the face of constant external perturbations? The answer is that they are responsive materials. As I mentioned previously, cells remodel their membrane compositions to maintain constant biophysical properties in response to perturbations. So far, this ability of homeoviscous adaptation has been only studied in organisms which cannot regulate their body temperature. But does this happen in mammals who can regulate their body temperature, but do have variations in their diet to deal with? When we fed mammalian cells DHA, a fatty acid which should have led to an increase in membrane fluidity, we instead saw that the membrane fluidity was unchanged. This suggests that the dramatic changes we saw from the lipidomic data resulting from the incorporation of DHA into the membrane lipids, specifically the increase in lipids with that decreased membrane fluidity, like the saturated lipids, are consistent with lipidomic remodeling for the purpose of homeostatic maintenance of membrane physical properties. However, the acyl chain remodeling in response to PUFA supplementation was incomplete, as the unsaturation index of PUFA supplemented cells remained higher than those from untreated controls. If the lipidome response we observed were designed to normalize membrane properties, the acyl chain remodeling appears insufficient on its own. So in contrast to prokaryotes, where homeostatic membrane adaptation by saturation of lipids has been well studied, membrane properties in more complex organisms are regulated also to a very significant extent by sterols. Thus, we hypothesized that the adaptive response to PUFA perturbation in mammalian cells was also mediated by cholesterol. So when we again looked at lipids isolated from either the fish oil-fed mice or a cell culture model of RBL cells, we found that cholesterol significantly increased with omega-3 fatty acid incorporation into the cell's lipidomes. In mice, we saw this both in lipids isolated from the hearts and the livers. Furthermore, in RBL cells, we could look at the time course of addition to cholesterol to the cell's membranes, and we found it to be extremely rapid. These observations suggest that cholesterol upregulation is an early and potent response to membrane perturbations. To determine how cells might be doing this, we looked at a main source of cholesterol addition to cellular membranes, which is its synthesis via the transcriptional activity of steroid regulatory element binding proteins, or SREBP. SREBP is a master regulator of cholesterol and fatty acid production through its transcription of genes involved in their synthesis. For SREBP to be active, it travels from the ER, where it recognizes membrane composition, to the Golgi, where it is cleaved in series by two different proteases. The cleavage of these proteases releases the cytosolic mature transcription factor of SREBP, which travels to the nucleus and turns on gene transcription. When we looked at SREBP proteolytic processing via Western blotting, we found that DHA mediates a fast and potent upregulation of the cytosolic cleaved fragment of SREBP, suggesting that SREBP is involved in this role of upregulation of cholesterol synthesis. To validate the role of SREBP in the DHA-mediated increase in cholesterol levels, we used a cell line which is deficient in its ability to regulate SREBP in response to lipid challenges. These cells are specifically lacking the Cyt1 protease. In the wild-type cells, which are now CHO cells, we see again that the addition of DHA to the cell culture medium leads to a significant increase in cholesterol in the cell's membranes, whereas cells which are lacking the Cyt1 protease and cannot respond via SREBP signaling, the cholesterol levels remain about the same as they were in untreated cells. Additionally, we can pharmacologically inhibit SREBP signaling by the action of the drug betalin. Betalin blocks the trafficking of SREBP from the ER to the Golgi, and thus again stops the signaling via SREBP processing. Again, in DHA treated cells, we see that cholesterol is significantly increased in the membranes of these cells, whereas in cells which were fed DHA in the presence of SREBP blocking by pharmacological inhibitor betalin, we see that cholesterol remains lower in these cells. And these suggest that SREBP is at least in part regulating the cholesterol addition to the cells in response to DHA. To test the idea that lipidomic remodeling is in line with maintaining constant or homeostatic membrane physical properties, we measured the general lipid packing in our cells. To do this, we used the dye C-Lardan and did spectral imaging to determine the membrane lipid packing, or roughly fluidity in cells treated with DHA for various times. C-Lardan is a somatachromic dye whose spectra shifts dependent on the presence of water in the membrane. From the spectral changes, one can determine the generalized polarization, or the GP value, which can be used to report the degree of lipid packing. A higher GP indicates more tightly packed, and lower GP means less packed or more fluid. Shortly after the introduction of DHA to our cells, we see that there's an increase in the fluidity or a decrease in the GP of the cell's inner membranes, denoted here by the white mark around the inside of the cells. When we inhibited the ability for the cells to remodel their membranes through the addition of betalin, we see that the GP stays low or the fluidity stays high. This response does not come back to normal after 24 hours as it did in the internal membranes. Furthermore, when we looked at the plasma, the whole cells through doing GP spectroscopy, we see again that in the presence of betalin, the GP stays low, or that the overall cell fluidity is higher and when the cells are unable to mount a lipidomic remodeling response. And we see this both with the pharmacological inhibitor and with the genetic blockage of SREBP signaling. Furthermore, when we masked the plasma membrane of these cells and looked at the changes in C-Lardan spectra, we saw that the plasma membrane is significantly more fluid in the cells in which were fed DHA in the presence of betalin. To more precisely validate the effects of lipidomic remodeling on the plasma membrane itself, we employed another method which utilizes a fluorescent porter of membrane packing, specifically in the plasma membrane. Dye-4 integrates solely into the outer leaflet of the plasma membrane, and similar to C-Lardan, the photophysical characteristics of dye-4 are dependent on lipid packing, with fluorescence emission lifetime providing a particularly sensitive readout. In this assay, higher lifetimes are indicative of more tightly packed membranes, and lower lifetimes indicate more fluid membranes, as shown here by the imaging of this space-separated giant unilamellar vesicle. We tested the dye-4 lifetime on cells that were fed DHA in the presence or absence of betalin, and found that neither DHA nor betalin alone significantly affected dye-4 lifetime. However, plasma membrane packing was significantly reduced in RBL cells, which were fed DHA in the presence of the lipidomic remodeling inhibitor betalin. So although biophysical membrane perturbations likely have several distinct physiological effects, we consider the central role of the plasma membrane as a selective barrier. We hypothesized that the permeability of cell membranes would be increased by uncompensated incorporation of PUFAs into the plasma membrane. To test this, we adapted a classical cell viability assay that relies on the passive diffusion of the amphiphilic compound, FDA, across the cell's plasma membrane into the cytosol. Once in the cytoplasm, cellular hydrolases convert the FDA into fluorescent fluorescein, whose charge inhibits its diffusion back out of the cell. Because the catalytic conversion of FDA is highly efficient, the rate of fluorescence increase is a direct readout of FDA flux through the membrane. We observed a linear increase in fluorescence over at least the first 10 minutes of our measurements, consistent with a constant flux across the plasma membranes. The permeability can then be calculated from the flux using Fick's law. Consistent with our hypothesis, cells with uncompensated membrane perturbations, such as those cells which were treated with DHA in the presence of betalin, are more susceptible to passive permeation by FDA. If homeoviscous adaptation of maintaining membrane physical properties in a range compatible with life is necessary for mammalian cells, then we would predict that disruption of this homeostatic response would negatively affect cellular fitness under conditions of membrane stress, such as those induced by PUFA supplementation. To test this prediction, we evaluated the cytotoxicity of DHA under inhibition of the membrane response machinery. At low doses, neither DHA nor betalin alone were significantly cytotoxic. However, DHA supplementation in the presence of betalin led to a 50% decrease in cell number after 3 days in culture. And we see again the same in wild-type cells, CHO cells. We again see that DHA is not toxic, whereas in those cells which are lacking the Site 1 protease, DHA is toxic even at low doses. To determine whether this was an effect on cellular proliferation or specifically on cellular death, we used flow cytometry to measure the proliferation rates of cells and found that the populations of cells at various parts of the cell cycle progression were unaffected by any of the treatments, suggesting that reduced proliferation rates are not the cause of lower cellular numbers. However, the combination of DHA and betalin, but neither treatment alone, significantly increased the proportion of dead cells, in this case indicated by trypan blue positive cells. We originally speculated that apoptosis may be involved in inducing cell death, perhaps resulting from an imbalanced signaling at the plasma membrane or membrane stress induced by lipidomic perturbations. One hallmark of apoptosis is the externalization of phosphatidylserine, which is marked by binding by an Annexin 5, without the loss of membrane integrity, which is marked by propydium iodide staining. So again, we use flow cytometry, and we can identify apoptotic cells as those which were Annexin 5 positive and PI negative. We did not see an increase in Annexin 5 positive PI negative cells in our cells which were treated with DHA in the presence of betalin. However, we did see an increase in the propydium iodide only cells. So the reduced viability that we see from uncompensated membrane perturbations appears to be due to non-apoptotic cytotoxicity. These results reinforce the idea that membrane adaptation or remodeling in response to dietary stresses is necessary for cellular fitness. Our data suggests that mammalian cells retain the capacity or homeoviscous adaptation for responding to membrane perturbations, though they have co-opted it for responding to dietary rather than thermal membrane stresses. So with that, I would like to end our presentation and thanking our lab and everyone who is involved in these studies.
Thank you very much, Kandice. That was brilliant. So I want to ask a couple of questions, a few questions from me and also a few questions from the audience.
Great.
So the first question I want to ask, so you're feeding the cells with a lot of unsaturated lipids, and you already showed that when you feed with palmitic acid or oleic acid, cells didn't like it, so didn't take it. So did you try to feed the cells with completely saturated lipids and check if it changes the unsaturated lipid levels?
So we tried feeding palmitic acid. We did not try feeding lipids themselves. We more fed the free fatty acids. And we've tried feeding palmitic acid, although only at low doses. When we feed them with higher doses, it's also toxic. But we have not completely followed up on that to be able to check to see if there's a compensation by increasing unsaturation.
Mm-hmm. So one of the audience questions, you partly probably answered it. Is specific lipid species uptake dependent on any membrane lipid composition sensors?
That's a good question. So that's kind of the next idea to test is what are the membrane sensors? What are the cells actually sensing? And yes, if they have a specific need for a particular membrane phenotype, are they going to specifically uptake? In this case, they seem to need PUFAs, and so they take it up. But yeah, I can imagine different cells in different conditions might have different needs. And that's kind of the next line, is to look for what property they're sensing and what specific proteins are involved in sensing that.
And related to this, did you check also some protein compositions if something changes in the plasma membrane?
In our previous studies, which we published in Science Advances 3 years ago now, we did proteomics and we did post-translational reverse phase protein arrays, where we looked at post-translational modification of proteins. And we found a lot of changes in the plasma membrane signaling, both growth factor receptor signaling, integrins, ECM. These were in mesenchymal stem cells, but I can imagine that this is the same in a lot of different cell types. So yes, I definitely think that changing plasma membrane lipid composition definitely affects cellular signaling, both at the plasma membrane and inside.
Okay, another question from the audience. What happens to the endomembrane lipid composition and homeostasis upon external lipid addition?
What happens to the endomembrane lipid composition?
So basically, you showed the GP for intracellular membranes, but did you also do the lipidomics for total cell and then deducted the endomembranes?
So far, we only have either whole cell membrane lipidomes or plasma membranes. And I would like to specifically look at what's happening in the endomembranes, such as the ER, where a lot of lipid synthesis is occurring. We haven't yet utilized, I hadn't quite trusted isolation protocols to very cleanly isolate ER or other membranes, although those have been recently developed. So that would also be something we're interested in doing next.
Okay, another question from the audience is, does the increase in cholesterol after DHA treatment return to the baseline with time?
Does the increase with DHA, so once we add DHA, does it come back to baseline over time?
So does the increase in cholesterol after DHA treatment return to the baseline with time? So you saw the GP returns to the baseline. Does the cholesterol level return?
The cholesterol level stays high. It stays, so in all of the cholesterol studies that I showed, those are, I think it's here, those are for mice, those are after 2 weeks of feeding. And in the RBL cells, that graph that I show on this slide is actually after 3 days. So they stay high, we believe, because the DHA also stays high. So it reaches a new set point.
Okay. So Pablo C says, I'm curious to know if cells respond in a similar manner to cholesterol treatment, increase or depletion. Would they just tune the cholesterol content or also respond adapting the lipid saturation levels?
I think that's a great question. And people haven't really done many comprehensive lipidomic analyses of animals such that say we're either had a high cholesterol diet or genetic modifies of the cholesterol synthesis pathways or treated with statins. So it's something that we haven't been able to really look at yet. Feeding in cells, we would need to also supplement with some of these PUFAs because the cells themselves can't make them. And so it gets a little more complicated in cell culture because they're reliant on what you feed them.
Mm-hmm.
But I can imagine, though, in a high cholesterol diet, that the cells would upregulate. In healthy animals, they would upregulate the PUFAs to compensate for this.
Yep. Another related question, does moderate DHA supplementation enhance viability? If so, does this suggest that these cells are PUFA deficient?
Yes, in most of the cells we saw, it seems that viability is slightly increased. And yes, I believe that all cells in culture are deficient in omega-3, specifically fatty acids. I mean, everyone we have ever fed, CHO, RBLs, fibroblasts, they all take up DHA very quickly as if they're almost starved for it and that they're not at their baseline. And so I do think that an appropriate amount in culture would actually enhance all of our cell culture studies.
So do you think we should also consider this in our normal cell culture to supplement it with?
I definitely think so. I mean, in neurons, it's a necessity that you have to have that in the cell culture media, some sort of polyunsaturated fatty acids.
Mm-hmm.
And I do think in cells, we've selected them to survive without it, but I do think that perhaps either it's more recapitulating normal in vivo conditions if you have PUFAs, or even just helps their survival, especially for the hard to grow cells.
Okay, David Christian asks, since SREBP is a transcription factor, have you considered doing RNA-seq of DHA-treated cell type to see if other pathways are also impacted?
I'm sorry, can you say that one more time?
So since SREBP is a transcription factor, have you considered doing RNA-seq of a DHA-treated cell type to see if other pathways are also influenced?
We have done some RNA-seq information. We did see the SREBP signaling was affected with RNA-seq. There were other pathways, but it was a little convoluted in that it really depends on the timing, I believe, of isolation of RNA. Are we looking at the initial response, or are we looking at the steady state once they've reached kind of a new membrane composition? And that's where we kind of left it and didn't include it in this manuscript, because it was tricky to figure out what point we were looking at. There are changes to different signaling pathways, mitochondrial signaling, and other lipid, fatty acid synthetic pathways were also involved. Mm-hmm. Different transcription factors were involved, but not necessarily like the RXRs or LXRs or any of those, which are also involved in fatty acid synthesis.
Okay. Considering diet, would there be a difference in the effect between plant-derived or animal-derived saturated fatty acids on cholesterol synthesis and membrane incorporation?
Plant-derived or animal-derived, keeping the unsaturation the same, I can't imagine that it would be different. But if we're talking about plant-derived having more omega-3 or omega-6 polyunsaturated fatty acids and animals having more saturated, then yes, I think it does matter where it comes from. But I think more it's the actual fatty acids you're adding that's the effect, not where it comes from per se.
Okay, the last question. These long-chain polyunsaturated fatty acids are highly prone to lipid peroxidation and can lead to cell death. Can you comment on how cells can regulate the amount of these PUFAs?
Yeah, that's a great question. We have always taken into account lipid peroxidation and lipid oxidation in these studies. As for how they regulate how much they're incorporating, such that they're not adding too much, and they can be more reactive oxygen species, that I guess it depends on knowing what the so-called sensor is for these properties, which I'm not sure of. I don't know if the cells just stop uptaking it, therefore they're not having additional in their membranes, or if they're uptaking it and specifically metabolically reducing the levels of it such that it's at safe levels. And that's not something that I know at this time.
So one more thing, I'm sure you get these questions a lot, because here what you're presenting is basically you feed the cells with polyunsaturated fatty acids, and overall the cell biophysical properties of the cell is the same. Is it still, if you think about the diet, it's still healthy to have polyunsaturated lipids, right? Even though anyway the cell balances out, you still have a lot of polyunsaturated lipids, although cells downregulate other polyunsaturated lipids. Is it correct?
Correct.
So basically still it holds that we are what we eat.
Definitely. And I know some people are shocked. We hear that excess cholesterol is bad, but yet when we feed polyunsaturated fatty acids, we're getting this huge increase in cholesterol. But I think it's truly a balance, and that it's the balance between the two that is changing our signaling, which is what's important really to study to figure out dietary effects. Because yeah, we are what we eat. If you eat a lot of fish, or you have a lot of fish lipids into you now.
Good, good. One last question from audience that just came. You mentioned about the changes in plasma membrane permeability after adding DHA. Did you check if there were any changes on viscosity? Also, is there any indication of spatial heterogeneity of these changes?
That's a great question. We haven't specifically looked at viscosity in these cells using any of the several sensors that could be out there. Looking at the spatial heterogeneity in our previous work, we have found that when you feed cells PUFAs and they have this increase in cholesterol and saturated lipids, that this specifically leads to an increased phase separation, at least in plasma membranes. And so they tend to have either larger or more stable lipid rafts or liquid ordered diptomates, and that this then changes possibly all downstream signaling. So we have seen the phase separation. Viscosity we have not looked at yet, though.
And what about aging? Do you think this balance is changing with aging?
That's a great question. I would have to say probably, but that's not something that I have really looked at in the literature.
But that would be a great question maybe for future, to get cells from old mice and young mice.
Definitely, and throw at them different diets to see how do they respond to it. Like maybe they can't respond as well in an aged animal versus not. I know aging in culture, not aging per se, but time in culture does definitely affect it, just because freshly isolated cells are very different from those which are have been cultured over time.
Thank you very much, Kandice. That was fantastic. Really loved it. Just mostly because I got to ask all the questions. So thanks a lot for your time.
Thank you, Erdich.